Okay, it's my great pleasure to introduce Eduardo Perozo. He's originally from Venezuela, where he did his undergraduate work. Then he did his Ph.D. work and short postdoc at UCLA. He came back. He did also, I think, a short postdoc in Venezuela. Shortly after that, he came back to the U.S. Accepted faculty position at the University of Virginia. 2006, he moved to the University of Chicago, where he is now. So I've known him for many years now. And every few years, he just publishes some fundamental cornerstone paper in the field of bacterial mechanosensitive channels and again revolutionizes the field. And he's done this again recently. But I want to point out this is not unique for the mechanosensitive channel field. I mean, he's been doing this for KCSA and HERGs and CATs and Core and many other channels where he's been just kind of turning over rocks and such like that, always using cutting edge biophysical approaches and such. And today his talk is entitled "The Structural Basis of Force from Lipid Gating in Mechanosensitive Channels." And so with that, I'll let you do your screen share and I'll go into the background.
Well, all right. Thank you. Thank you very much, Paul. It's the check is in the mail, by the way. You don't have to say such nice things because now, when you get the strong introductions, the seminars are usually disappointing. But anyway, I will answer one question which has already came up in the chat, and Ryan wants to know what this is. This is actually Therodon dinosaur print, which you can actually go and copy yourself in Connecticut dinosaur tracks park. So I have a weakness for fossil hunting, and so there you go. So I am going to share my screen here. Here. Do you guys see this? I hope so. All right. So let me get on.
Yes,
okay, thank you.
Perfect.
All right. So let me get on going. But before I start, I want to highlight the fact of the people that actually did the work. And this has been a few years of a very lovely and very satisfying interaction with 3 people in the lab. But the driving force of this has been Bharat Reddy, who had been converted into the mechanosensitive channel cult, and he really enjoys it, and he's responsible for most of the cryo-EM data that I'm going to show, and together with Navid Babi, who is an ex-Boris Martinek, so it's a nephew of Paul, I guess, scientific nephew, and he's also obviously contributed with electrophysiology and computational analysis, together with bunch of people in the lab. This is a very open project. And so I probably don't need to do this, but I always like to start by setting the stage, by calibrating people in terms of the energetics of the systems that were involved. And for those of you that know me, my main interest in science has been always the issues of how energy gets transduced in biological systems, and how do we address these energy transductions to produce function, which in case of signaling, in most cases, signaling or response to sensor systems, regards the transduction of multiple forms: voltage, ligand binding energies, force itself, light, temperature, into ultimately protein motion. But the problem of addressing this issue when we talk about mechanobiology and force transduction is that not only there is a huge dynamic range. We go from the truly most of a very blunt instrument, which is how to respond to large osmotic differences, for instance in bacteria, which we're dealing with 10 to the 5 newtons per meter square. to faint sounds, which are almost 8 to 9 orders of magnitude weaker sort of forces. But the problem here is not only the range of energies that are involved, which necessitates many different evolutionary solutions to the problem, but also the fact that because we're dealing with force, which is a very extensive type of a mode of energy, in principle, all molecules should be force sensitive. And in fact, they are. I mean, the issue of what is a mechanosensitive system and what constitutes a mechanobiologically interesting principle goes to the fact that if we had enough force and a small enough probe, you probe a molecule, it will respond by force. And so I give you a couple of examples. One is, we all know that shaker is a classical voltage-dependent ion channel. Nobody thinks of it as a mechanosensor. But in fact, if you pre-activate shaker and look at single-channel traces, and this is work by Kathy Morris a number of years ago, you actually modulate the way in which voltage gets affected by it. But that doesn't mean that shaker is a mechanosensor. This is different between physiology and biophysics. And by the same token, we can involve energies or forces that are so large as to promote non-physiological events like protein unfolding. And so this is a classical example of force spectroscopy using a cantilever-based atomic force microscopy, where we can actually link titin, which is a structural protein in muscle that has many repeats. And if we apply enough force, as I said, and with enough small enough probe, we can actually measure the process of unfolding individual. So what we're going to be dealing with here relates more of the transitions that I explained before, and it had to do with the fact that work originally in the Coons lab made the major discovery that there are these mechanosensitive ion channels in prokaryotes. And these mechanosensitive channels have become sort of the most studied, the standard bearers of the development of most of the ideas that we know to the process of mechanosensitive gating. The process is physiologically speaking, real. It's part of the osmotic response to challenges in bacteria. The vast majority of bacteria have them. And this is one example where you can activate the channel by applying transbilateral pressure, which then transduces into in-plane membrane tension, which then has an obvious dependence with the open probability. We can see here. This probability relates and has been suggested for a long time. People like Paul, Boris Martinak, Sergei Sukharev, Jim Kuhn have actually placed the foundations of the field in very solid grounds. And the very basic idea is that if we model this like a little cartoon, and we have this delta force or tension in this case, the idea is that we have some sort of spring attached to a gate. But in reality, this delta force can be explained by a very simple equation in which the tension and the mechanical dependence of the gating relates to whether or not there are changes in the cross-section area of the channel. So this sort of sets the base for what we're looking for in terms of structural biology. But in fact, not only this channel has been cloned and the structure known for a long time, but also a number of other mechanosensitive channels have come to the fore. And I'll give you a few examples based on the basic principles that we now understand drive the gating of these channels. On one side, we have the concept of force-form-Lipid, which is either direct tension, the hydrophobic mismatch, energetic differences, or what the physics call lipid curvature frustrations, which is when the membrane is naturally curved and you oppose that, there's a delta force that transduces in the plane of the membranes of tension, which tend to then open the channel. We and others have looked into this experimentally, and this is a fairly solid concept in the field. On the other side, there is the original idea that perhaps these channels are linked to cytoskeletal elements, and this process leads to either indirect or direct links from the intracellular cytoskeleton or the extracellular matrix. And for that, we have only one true bona fide example, which is a trip channel, NOMC in Drosophila, that actually has been shown to, or suggested that is responsible for hearing in insects, in arthropods in general. Interestingly, my life is divided into my interest in voltage sensing and the driving of conformational changes by harnessing the energy in the electric field versus mechanobiology, which is the topic today. In the voltage-dependent field, the major biophysical problems that evolution sort of dealt with were solved a long time ago. So for that reason, if you take a bacterial potassium channel, it's the way it selects ions is very similar to the one we have in our brains, or the way the voltage sensors tend to work. They're also very similar. So these were problems that were solved, and they're so efficiently solved that the fundamental basis of the evolutionary solution to these problems have been kept for a long time. That's fantastic because you can use these prokaryotic systems as model systems to address structure, function, a number of ways. And of course, I don't mean to suggest that studying eukaryotic systems is not the way to go. In fact, it is the way to go because there are much more nuanced and tweaks to the regulation that define our physiology, not bacterial physiology. But the point is that this makes it fairly mature in terms of the different forms and dynamics that these channels take. The mechanosensitive channel field is not that way because there are multiple ways in which force can be transduced. And this is very often the case by the fact that we now have a wide variety of different channels or structures or design blueprints that all respond to different types of forces in the cell. And so, fortunately, we have now a number of those at atomic resolution or quasi-atomic resolution. And today we're going to talk about one in particular, MSCS. But there is a very rich literature in MSCL, and of course, piezo's and ENAC type channels, tracks are all involved in mechanobiology in eukaryotic systems. So let's see. So what did it say? Leo Tolstoy suggests that War and Peace that all happy families are boring because they're more or less the same. It's the unhappy families that are unhappy in their own little way, and this is exactly the case for mechanosensitive channels. So, oops, sorry, go back. There you go. So let's look into the forces that we have been dealing with in this many years. And by fact, what I'm doing here, it may be known to you, but I want to set up the state of the field before I make a transition to our current work in the lab, and actually others in the field as well. So what sort of forces are we dealing with when we look into the types of conformational transitions that open and close a mechanosensitive channel? In the classic work by Cantor, you can get a trans-bilayer profile, which is informed by both the interaction at the point of juncture between the phosphate group, the glycerol and the azo chains, and the dipole potential in it with very large attractive and repulsive forces. In addition to van der Waals matches and mismatches in the very mobile azo chains. And it gives you this weird looking pressure profile. And we can think of a couple of ways in which you can move that. You can change this profile by modifying the footprint of the membrane into the channel and vice versa. And this we refer to as the hydrophobic mismatch energetic differences. Or, as I said before, you can have curvature frustration events, which are very interesting to deal with because you can change the tensions in the bilayer by modifying the composition of the membrane using fairly basic geometric principles. And so, for instance, we know for a fact that the forces in the bilayer and the intrinsic curvature are defined by the geometry, the overall geometry of the individual components in the bilayer. So the classic example is sort of PC, where the cross-sectional area of the head group is roughly the same area as the cross-sectional area of the acyl chain, and that will predict a cylinder shape, which will form a fairly flat lipid bilayer in two dimensions. The moment you're dealing with cone-shaped lipids, either detergent type cones, which have this positive curvature, or you can have phospholipids like phosphatidylethanolamine, which has the opposite orientation or geometry, you'll get two types of distortions, and both of those lead to curvature effects. And these curvatures, if opposed, then we'll have a membrane, intramembrane component of tension, which then affects ion channels. So having said that, let me review a little bit of what we know, one of these examples. And, this is in mechanosensitive channels, as you probably know from the work of Paul. In the prokaryotic systems are fairly divergent and convergent, and then many, many different examples of two classes. The MSCL type, which is a large conductance, but it's a tiny channel. It's a small pentamer with just two transmembrane segments. And then you have MSCS for the small conductance, but turns out to be a larger molecule, which is a heptamer. And this is a structure as obtained by the lab of Doug Rees almost 20 years ago, which is amazing. So whenever you see this, and when we saw this, everybody in the field said, this is fantastic. Look, we can already see where the membrane should be. There are in principle, three transmembrane segments, or maybe 2.5. This is partially in, and it forms a pseudo-symmetric or symmetric system where you have a fairly large opening. But the walls of this permeation path are highly hydrophobic. So there are all these interesting juxtaposition between physical blocking the conductance and a dielectric pore, which Sergei Sukharev looked into the wetting mechanism, where you have a little vacuum. Is so hydrophobic that water and the walls so restricted that even though the geometry is enough to allow ions to go through, they don't, because there's no water in it. And so this de-wetting transition has been at the center of understanding opening and close once you open much larger diameters. The other interesting thing is the fact that if you look at a surface representation of the structure, not only you see that within the putative transmembrane segments, you have these cavities here, which are also hydrophobic and have been proposed by a number of people, Cleodas et al., Booth and Naismith, have been suggested that this is actually filled with some sort of hydrophobic molecules or lipids. But the interesting thing is, in terms of these energetics of interactions between the membrane and the channel, and by the way, I mean, I usually say this at the beginning, but one of the interesting things about these systems is that the physiology of the channel is defined by its protein-to-lipid interaction. So the interface between the membrane and the channel plays a fundamental role in understanding what might be going on here. So among the first computational analysis based on the structure pointed out that the putative transmembrane segments have this trapezoid architecture. And then if you place this on a lipid bilayer, the lipid bilayer will actually distort somewhat. And this distortion might play an important role in not only interaction, but the transmission of forces into this protein to lipid interface. And so I'll leave that to you to brew a little bit. Then a while ago, first in collaboration with Boris Martinak, and then with Valeria Vasquez, who now has her own very successful lab in Tennessee, we did a series of experiments to actually quantify the whether or not we could use this curvature frustration energies to actually open the channel in vitro and in the absence of any tension. Because the idea was at the time, since we were doing a lot of spectroscopic work, how do we actually, the challenge was how do we address this with some structural information to see what parts are moving, how do they move, and how do we address the modeling of the open and closing transition. And so this is one of my favorite experiments where you first reconstitute the channel in a liposome, patch it, excise the patch, and then apply tension, as many people in the field have done, and then you see, some sort of activity. Then we end the pulse of the pressure pulse, and then in the nominal absence of any tension, then you apply a cone-shaped lipid that incorporates only in one of the leaflets. In this case, it would be lysophosphatidyl-choline, which is a cone-shaped 18-carbon length amphiphat. And if you do nothing but just wait to see, you see an initial activity, and then all of a sudden you see macroscopic currents. You have they tend to stabilize at a stable rate of incorporation and exit of the lyso-PC into the patch. And then you add a little more, and you recruit even more of these ion channels. And the patch always ends the same way. It blows up because there's always more lysophosphatidyl-choline to enter this and destabilize the mechanics of the bilayer. We've done this a number of times. And with this, we were able to, using electromagnetic resonance spectroscopy, go residue by residue and look at the conformational differences in terms of distances and accessibilities to produce models. At the same time, this was published, Booth, Naismith, and collaborators were able to solve the crystal structure of a putatively open MSCS. And indeed, this was done by using a gain-of-function mutant at position 106, which is at the very close to the narrowest portion of the permeation path. And it gave us the first very much interesting view of what might be going on. And this confirmed a number of the earlier proposals, which suggested in order to open the channel, we needed to have increase in the tilts of the alpha helices present already in the supposed closed or the desensitized state. We did the same using more modeling than direct structural determinations, modeling all of the EPR before and after adding lysophosphatidyl-choline. And they're not identical, but the idea is similar. You get an expansion to about 11 to 12 angstroms in diameter, and that increases the tilt of individual helices. And at the time, this is 10 years ago, we were 11 years ago, we were fairly satisfied. Oh, this is an interesting hypothesis, and it makes sense in terms of what we know on the function. And so let me then summarize, based on this, there are three major ideas underlying the mechanisms of how this channel senses forces and then opens. And in fact, credit to where credit is due, the first credible sort of physical idea that suggested how these types of channels open actually came from Paul. And he called them, or Paul and collaborators, Marr and Elson. And the idea was that perhaps what's happening is that the membrane itself exerts stabilizing forces. And once you apply tension, which decreases the lateral force, those forces get counteracted, and now the channel jumps into the open state the way a jack in the box will, go out a box. And so that's called the Jack-in-the-Box hypothesis, and assume that there were intrinsic energies in the child. That turned out not to be because, well, you can actually stabilize what appears to be a non-conductive conformation in the presence of detergent, and in detergent micelles, the forces involved in the bilayer are clearly not the same. So the other two ideas are more of a allosteric sort of interaction between the different parts of the transmembrane segments and the membrane. And we call these gating through interhelical interactions. Or the idea that in this cavity that I mentioned before, the lipid itself stabilizes the closed conformation, and once you apply tension, you release that, the lipid acts as a ligand, and the ligand leaves that cavity, thereby opening. This has been bolstered not only by seeing some lipids in the cavity, although at the time it wasn't clear if it was lipid or detergent, but also work by Pleotas in collaboration with Naismith, Booth, and Carl Robinson show that in fact, if you purify native MS from native membranes, you will get a number of different lipids already bound to it. And so that was the state of the art a few years ago, not that many years. And that's when we decided to get back into the question of this after stopping for a little while. And the basic idea was, well, now that we have cryo-electron microscopy and we can actually address this, perhaps we could look into the influence of the bilayer itself, and maybe we could see some of these lipids as the driving force for gating. So allow me, if you will, for a moment, to give a big detour. And I want to talk about psychology. And I want to talk to you about cognitive bias. And the reason I'm saying this is because with the results I'm going to tell you now, I found myself going back to our previous interpretations and realizing that we're all really susceptible to this. I mean, all humans are cognitive bias in a way or another. But I'd like to highlight a few of the things that I felt I was guilty of. And it affected the way we not only interpret data then, but now how we interpret data now. So, you know, clearly the cognitive biases tends to affect your rational scaffold of, you know, how do you address information, how you interpret information. And so there's 3 or 4 ones here that, as a scientist, we really need to get into. One is confirmation bias, which is that we tend to favor more the data that confirms our pre-existing notions than things that challenged. And so I leave that for the moment there. Then there is the anchoring effect, meaning if you see something first, that stays far longer than anything that comes in afterward in terms of new data. But my favorite is the survivorship bias, which came to fore after World War II, when the research of Abraham Wald, a statistician for the US Army, at the time showed a remarkable example of what this might be. The statisticians were tasked with the job of addressing how to best protect bombers during day raids in Europe. And they came out with all this information of every single hit that all of this in aggregate were suffering. And you can see here in the plane where these are. And the general said right away, aha, we need to put extra plates here, 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 and here. And Wald, after looking at the data, said, you have it 180 degrees wrong, because the results of the data comes from only the planes that came back. The ones that were really affected never came back. And in fact, these are these empty spots right here. And that's where the armor went. And of course, the bandwagon bias, which is what leads us to do groupthink. Now, the reason I'm telling you all of this is because when we looked at the crystal structure of MSCS, which was a momentous achievement for the field, we see not only an obvious location for the bilayer, but also the fact that there are regions that are not included in this structure, and these regions we dismiss as they're not that important, they're probably not folded, they're too dynamic, mobile, et cetera, et cetera, et cetera. And in fact, when you look at the crystal contacts from the Bass et al. Structure, you see that the entirety of the crystal is supported by interactions between the most stable, certainly stable part of the channel, which is its cytoplasmic domain. In the transmembrane segments, and presumably the micelle is completely excluded from any contact that could actually stabilize it. So based on that, Bharat, when joined the lab a few years ago, I suggested, why don't we take a look, since you want to try cryo-EM in membranes, in nanodiscs, why don't we try addressing this question? And at the time, it was just simply a test, and a control experiment, if you will. Well, long story short, we reconstituted an MSCS in nanodisc. The biochemistry has been robust for many years. And we found conditions, even though this is a little bit orientational bias, you can address that with either tilting of the grid, or what we ended up doing was doping the grids with fluorinated detergents, which tend to exclude a fraction of the channels from the water-air interface. And this is apparent here. When we look at classification, we basically see two classes. One where there's no transmembrane segment, and this is what we think is at the membrane air interface, and one that gave us the actual structure. And right now we have a variety of structures, different resolutions, but this is reliable and it works really well at around 3 angstroms, sometimes less, 3.5. It's sort of the classic range. And as you can see here from the local resolution maps, the N-terminus, and I'll talk about that in a second, is not only there, but this hairpin between TM2 and TM3 are the most dynamic, most mobile parts of the molecule. So let me then redress this now with the cryo-EM structure in a lipid bilayer. One thing jumps to fore, which is one, that there's a new component, there's a new domain of the channel, which we call the anchor domain, and it extends TM1 2 or 3, three actually, in terms of a helix, and it forms this little loosely interacting anchor, which also goes back into the membrane. So that's one thing. The other thing is that when you compare that to the crystal structure, anything below this new part is essentially identical to the crystal structure. And that means that the narrowest portion of the permeation path continues to be this L105, and that the balloon that I showed you before is also quite stable and contains these fenestrations, which make this very large water field cavity. So looking into this, and then I'll give you a little flyby. We're taking out the membrane part, and when we look at this domain, this is where the membrane interface is, and I'll go into the membrane aspects of it in a second. But as we do that, we can see sort of a re-entering sort of a globular mini domain, and they interact with each other right at the interface. So there are intercellular interactions that stabilize this, and of course there are intra-globular interactions, which are highlighted here. In addition to that, one of the interesting aspects of MSC has always been that, as opposed to the vast majority of membrane proteins, there were no obvious tryptophans that form the tryptophan belt that all membrane proteins have. And in fact, what happens is that this was part of an unfolded region, and when you saw the structure in the membrane, you see that there is a tryptophan firmly involved in a protein-lipid interaction. Um, so let's look into how important this N-terminal domain might be, because early work by Booth, in the absence of any structure, they did some fairly uh, blunt deletions to see which parts were important. And if you cut most of what you see here in the TM1 and N-terminus, it's not functional, but it was assumed that the membrane interacted with TM1 and the N-terminus was completely unfolded. And so here's an example of reconstituted MSCS, and this is standard. You give it a ramp, and you see the activation and the deactivation in the same experiment. If you then make our biochemical prep, which has a histidine tag in the N-terminus, it turns out that you get an additional sort of structural element here. I've never seen this before, but the histag forms a stabilizing zipper, if you will, which I think, no question, it helped us resolve the structure of the N-terminus itself. However, that construct is thoroughly is dead. It's non-functional whatsoever. And if we do an experiment in which we add thrombin to this media, it cuts this, and we can confirm that, and now it becomes active. And so that means that there's an interesting energetic aspect of the stabilization of this that stabilizes the closed state, but it is unable to allow the channel to open. And so out of sort of curiosity, even though the construct remains the same as that of the Doug Reese's structure, and it contains the N-terminus, we just got rid of everything that we don't see in the crystal structure, and that too is non-functional. Alan Liu, which is talented undergrad in the lab, then went on to do a systematic alanine scan of every single residue in these new regions of the molecule. And we show that, in fact, by doing downshock assays of bacterial survival, we see that physiologically speaking, alanine mutations to about 75% of all of these residues turn out to be loss of function. And when you map those, the worst aspects, the worst effects are both at the subunit interface, which are all located here, and in the membrane protein interface, which are located here. And that makes it a very compelling suggestion that we need to reassess the physiologically important ingating of this new domain. So I keep repeating the aspects of look at where the membrane is and take a look at how the N-terminus interacts with the membrane. And so perhaps our biggest shock was when we looked in globally where the nanodisk is located. And by the way, this nanodisk, what we see here in density is not the entire nanodisk, because the averaging that we use for a single particle reconstruction centers on the channel itself, but the channel itself might be moving around in this nanodisk. Therefore, the peripheral will all average out, and we only see a little bit of it, but enough to see where the membrane is. And the membrane turns out to be a full. So this is where the Doug Reese's structure predicts logically, and we and everybody else in the field thought the same. It predicts where the membrane is bound, but in comparison, the reality of it is that this is displaced about 12 angstroms. It's almost a full bilayer, or at least the dielectric, half of the dielectric, low dielectric of the membrane. So this is important for many reasons. One, A, the membrane is not the membrane forces are not applied where we thought they were. There's significant shift. B, the interactions of the membrane also favor, as I said, the confirmation of this new physiologically important domain. And C, everything that we've done before probably needs to be repeated in terms of computations, because as I said earlier, the forces are not where we think they are. And we also solved the structure of the delta, the deletion mutants. If we get rid of all these, the structure is not high resolution, but we can see that the bilayer itself remains exactly in the same location as before. So there's a number of ways in which we know this is relevant, physiologically relevant. Number one, we can change the composition of lipids and the location of the membrane stays the same. These are different structures that we've done in either POPC, POPG to mimic the E. coli lipids better than I'm sorry, this is in the absence of the histone. Sorry, in the absence of histone, it remains the same. But we can also do POPC, POPE as the basis of E. coli lipids, and the membrane remains exactly in the same location. And if we do it in DDM, the micelle stays exactly in the same location as the membrane. So that is all consistent. The second thing that we did was computational analysis of where the membrane is. And so we did a free energy analysis of the system when the membrane was at different locations. And we moved the membrane along the z-axis. And it turns out, not surprisingly, that the lowest energy system corresponds to the location of the membrane, that the cryo-EM structure tells us. And even if we start in different locations for the membrane, and we let the system evolve in molecular dynamic situation, they all converge to the same location of the membrane, which happens again to be the cryo-EM. So here's where things get interesting. As the resolution of our structure got better, and now we have this sometimes sub-3 angstrom map, we could see that there were actual densities that correspond to potential lipids. Two to highlight. One is what we call the pore lipid, which sits right in the permeation path, and we'll talk about it in a second. But by far most importantly is the presence of this density here, what we call the hook lipid. It's a clear phospholipid that embeds its head in the cavity that's formed by the top of the TM3 and the hairpin with TM2. And that has been always an interesting sort of cavity that people have indirectly talked about, but nobody took it seriously. So let me bypass this for a second. So going into this hook lipid, we can actually see that the head group goes into the permeation path, and the acyl chains are all placed away towards where the membrane is. And what we think is that this hook lipid is the actual interface between the channel and the membrane, and it may be the fulcrum of the transduction of energy, or at least an important component of transduction of energy. In addition to that, there are the lipids which we don't see in our structures. There are the lipids at this cavity that may actually interact indirectly. But if you think about where the membrane is, this doesn't correspond neither to the outer lipid nor the inner lipid, and is what I call a truly bound lipid as opposed to an absorbed in the surface. So we carried out initial molecular dynamic simulations where we have our system, the way you see it here, and we then computationally eliminated that density. And two runs, one were without this bound lipid, and another one with the hook lipid involved. And I want to show you the analysis. In one case, we apply enough tension to produce an expansion of the periphery of the channel, but at no point the permeation path is open large enough to be able to allow ions to go through. But this is not the case for the hook lipid. The hook lipid appears to make the expansion, then transduce into an opening large enough to produce activation. And you'll see it at the very end. Here, it open up. And we can see this in more detail here when we look at the evolution of the peripheral distance versus the pore distance. The periphery expands, although it expands more efficiently with the hook lipid. But although it tends to open only slightly, it truly opens about 3 or 4 times larger when you include the lipid. And so we went back to our data. We have extensive EPR data on this. And it turns out that we had this, and this is where some of the biases come in that I mentioned before. We had this all along. When you map the old EPR data from Valeria, into the new structure, they all fit not only perfectly, but the distribution of accessibilities match what you would expect is the exposure of the N-terminal domain and the very tiny cavity present to the external solution. So I'm finishing in a couple slides. And the basic idea is everything that we thought before in terms of conformational transitions in the modeling underlying this needs to be reassessed. Neither this allosteric model is correct because the structures underlying it are not correct. But I don't think that the exchange of the cavity lipid is enough to generate a ligand binding model of MSCS in itself. Instead, we have two options where we highlight not only the lipid that is in the cavity, but the hook lipid as a key participant in the forced transduction of MSCS. So at the moment, we cannot distinguish between two options. One is that you can consider in both options, you can consider the hook lipid as the old, stiff shift cars where you need a clutch to link the engine with the transmission. And so in this case, the engine would be the forces in the membrane, and the transmission will be the conformational changes in the channel that that produce an opening. So in model A, the bound lipid, the hook lipid, remains attached to it no matter what. And therefore, this is more of a allosteric based model where forces get transduced and communicated through the presence of the lipid into the interface between TM1, TM2, and TM3. By the way, TM3 needs to be reassessed because, in fact, given the location of the membrane, it's actually never in the membrane. The gate of this channel is cytoplasmic, it turns out. And the second model assumes a hybrid with the ligand-gated model where the conformational transition driven by these leads to the hook lipid to dissociate, and then that opens. But in order to close, the channel needs to rebind the hook lipid again. So in conclusion, we now have a new perspective of how the channel might gate, informed by everything that we saw before, but in the new context of what the membrane is, where the membrane is, and how forces may be transduced. So I'd like to close by saying that, just like the ribosome is a ribonucleoprotein enzyme, I'd like you to think of MSCS as a lipoprotein channel that you cannot understand only from the point of view of the protein, or only from the point of view of the lipid. And with that, I thank you very much for your attention, and I'm open to questions.
Okay, I will ask people to write their questions either in the Q&A or raise their hand, or they can even do it through the chat if they like. I guess I'm going to start by asking, I know some examples where, I don't think that they call them hook lipids, but there are some other channels where
absolutely,
where
yes.
lipids, and also not just lipids along the top and sides and stuff, but I'm also curious, are there other examples of lipids within the pore, and can you go a little bit more into detail about that?
Yes, I ran out of time, unfortunately, and I wanted to talk a little more about the pore lipids. And this is where the reason why I didn't go into it, because that's actually where we are the fuzziest in terms of we don't have a complete picture. It is clear that no matter what, no matter the condition that we solve the structure, as long as you solve it in the membrane, we always see what it looks like acyl chains. We have never seen the head group. And we don't really have a pathway for it to disappear or to move away, but it is clear that in order to open and conduct, these lipids need to move away from it. That's one thing. The crystal structures, none of the crystal structures have any density about it, but we actually went back into the original electron densities from the highest resolution Doug Rees's structure, And we actually see, let me see if I can share that with you now. Share screen. I don't have time to go into that, but there we go. We can actually see in the X-ray density one blob here that was never interpreted because it's uninterpretable, but is exactly in the position of the most electron-dense atom in the phospholipid, which is the phosphate group. So I know that that's not a demonstration, but it actually is tantalizing and is consistent with. Now, in regards to your question about other examples of lipid involved in mechanosensation, the best known example is the TREC channels that Brohan and McKenna have seen. The acyl chain, there's a canal from a cavity, a hydrophobic cavity, towards the pore. And their assumption has always been that as the channel gates, that interaction between the lipid and the channel drives either gating or has to move away to allow for conductance. I know that answers the question.
Okay, so there are a couple of other questions, or I guess Dr. Gloss has great talk. I agree very much with the lipoprotein ion channel idea. And then someone asked, does the A106V mutants that discussed produce an open conformation within the cryo-EM? And I'm actually interested in this one too, because
the answer is yes. We have that structure now, and we're actually finishing up manuscript, but that wasn't ready for B2. It's not fully close, but there are differences between the EM and the X-ray structure, mostly because the bilayer itself now stabilizes. So the main change goes very much along the model that I put in, the cartoon that I put in at the end, which is that the N-terminus transforms or becomes an extension of TM1. And TM1 it tilts significantly into the bilayer. And we saw that by EPR many, many years ago, which suggested something, but because the bilayer was not in the right place, we didn't interpret it correctly at the time. But we saw that data, and we have the delta accessibility, which means we know that the accessibility to the external solution of the N-terminal domain goes significantly down in the open state, consistent with the fact that it becomes part of TM1.
Okay. I'm going to open a question from Claudio Catalano. Let's see, I'm going to allow him to talk, so you should be able to open the mic.
Can you hear me?
I can hear you.
Yes. So I have a couple of questions. How do you compare your structures with the one from Rasmussen?
It's identical structures, except by ours, Rasmussen didn't have the luck of having an internal histag. And so they decided, they went through a fairly elaborate process of stabilizing the nanodisk itself, because the problems of orientational preferences are serious. So what they did was to link several MSCS in tandem to be able to shield that from the water interface, which is how they got the structure. But the structure, their N-terminal structure is significantly less defined.
Yep. But
so in our case, we did that too. So we saw the structure in the absence of the histag, and it is there, but the resolution does go down.
Yes.
Oh, I'm sorry, I muted him. I can unmute him again if he has a follow-up.
Yes.
Okay.
You're muted, Claudio. There you go.
You hear me now?
Yes.
Yeah, okay. They saw lipids in the interface between the TM3B and...
that is correct. We didn't. The main difference is that they made their grids at 4 degrees. So that we might be, that's fascinating, because we might be seeing a phase transition problem.
Yeah.
or a phase of the issue, not a problem. It's not a problem. We don't see it at room temperature.
Okay. Another question here: would it be possible that there is a kind of preference of specific type of lipid that binds to the channels, and might that influence the location of the channel in the membrane?
I'm sorry. Could you repeat could you read the question? Sorry.
Okay. Would it be possible that there is a kind of preference of a specific lipid that
Yes.
binds to the channels that might
It's called selectivity in a way. Yeah. Absolutely. I think there is we were able to identify a number of specific interactions between the protein and the head group of the lipid. There are no specific interactions with the acyl chains. And in fact, one of our acyl chains is quite mobile, and we only see a little stump. So, but it's clear a phospholipid. Now, the nature of the chemical nature of it requires high resolution, and we could not distinguish whether it was PC or PE, but in 99%, I'm 99% sure it's PE, mostly because of the mass spec work of Carl Robinson.
Okay. And then finally, we have one more question over here. Again, Dr. Gua. My question is, how do you propose the existing state of the interlipids when the channel is open? Will then still there be a, oh gosh, will then still they be gone? Okay, will they still be there or will they be gone? So basically, how do you propose the existing state of the interlipids when the channel is open? Are they there or do they disappear?
The lipids in the cavity or the hook lipids?
I'm going to assume that he means in the cavity.
Yeah, so I cannot answer that.
Dr. Gua, do you want to raise your hand and ask your
That's right.
question?
Maybe that would be best.
That would be best. Go ahead and raise your hand if you know how to do that. There he is. Okay. You should be able to talk now.
There you go.
Okay. Can you hear me?
Yes.
Okay. So my question is the lipids within the channel. There's like
The pore lipid or the hook lipid?
not the hook lipid. It's
the pore lipid. Yes. Yes.
Lipid. So
how do they go in and out?
It's a fulfilled the channel.
Yes.
So when it open, will the lipid be gone or it still be there? Or?
Oh no, it has to be gone. I didn't go into the details, but we've done all sorts of calculations that in the presence of this lipid, not only you do not need a vapor lock as suggested by Zuckerman, because it's actually at the location of the vapor lock. But there is absolutely no water penetration at all, at least in this conformation.
Okay, so if they are gone, so the question is how they come back.
I have no idea.
That's mystery.
I have no idea. This I think I mentioned it before in the talk, but it may have gone too quickly. This is we describe what we see, and in this conformation, which I think corresponds to the closed state, we see this density, and this density is consistent. It's systematically present, no matter the conditions of the nanodisk. Now, one possibility which we discuss a little bit in the paper and we discuss among ourselves is perhaps that might be involved in the process of desensitization of the channel. As we didn't dealt with it extensively, but MSCS has this sometimes disputed event that, as you activate it, it sort of comes down in liposomes. Not so much in spheroplasts, but one possibility is that we're dealing with a reconstituted system and the lipids themselves go in and stop conduction.
Mm-hmm.
That's interesting because, yeah, it is actually, I think, Dr. Sugraff basically said that he distinguishes between inactivation and desensitization. Desensitization, I think, is
That's correct.
My interpretation is oftentimes a lipid phenomenon where you're
yes, absolutely,
and you're changing the elasticity of the
exactly.
but but inactivation, I think, is quite different, and we see it as a real phenomenon even in spheroplasts, where
I see.
if you do the ramps correctly or incorrectly,
Yeah.
the channels go away, and you have to basically take all pressure off of the patch for many seconds, like 90.
No, I completely agree. And one of the reasons why I was convinced that this might be, and that's why I call it desensitization, because it's an accommodation of the membrane around the channel, is that MSCL doesn't have it. It's never shown this type of accommodation. But MSCL opens at much, much higher tensions. And so that means that you're probably extending the bilayer to almost its limit, and there's no room for any accommodation, whereas this, which is lower tensions, may allow some accommodation. But then again, as I told Dr. Wu just now, I have no idea.
Yeah, well, the one comment I would make that would disagree, I think, with that particular hypothesis is that we can make mutants in the skull, which are much more sensitive, even more sensitive than the skus.
That is
They
true.
still don't show any desensitization. So it's probably a function, a larger function maybe of the protein.
Yeah.
But, I think Ryan Hibbs has a question here that may relate to this. Maybe the pore lipids are there as a function of extraction in the detergent before nanodisc reconstitution. So he's, I think, asking is it, well, I'll just repeat the question one more time. Are the pore lipids there just as a function of extraction in the detergent before nanodisc reconstitution?
That's an excellent question. And the answer is yes. Whenever, I mean, these are all, obviously, in order to put them in nanodiscs, you have to extract them first in detergent. So whatever the hook lipid is doing and whatever the pore lipid is doing, it comes during the purification. We don't do anything special. And for that reason, I think that the hook lipid is not that easily exchangeable. The main difference is that when you solve the structure in detergent, then the binding site of the hook lipid involves that cavity, which in the roof of the cavity is the N-terminus. So once the N-terminus unfolds, that binding site suffers and you don't see the hook lipid.
We have one more question with a hand up, so I'll allow that person to talk. Oh, it's done, I think. Yes?
Oh, there.
Don, how are you, man?
Hey, okay. Yeah, as a general perspective, I'd like you to comment, if possible, a little on how different the principles are in the best known mammalian mechanosensitive channels compared to the bacterial. So this is an old problem question.
Yes.
I would like to hear your perspective on how all of the work on the bacterial structures and principles apply or do not apply to the mammalian.
Yeah. No, that's a great question.
Piezos and so forth, starting with the amount of tension needed to open.
Well, the amount of tension is slightly different than the fundamental principle. The amount of tension is the way that you can have, different types of the same type of sensor responding midpoint of activation. It's when the transition occurs. And it has to do with how deep the well and the peak of the energy landscapes are. Having said that, there is a clear correlation between the degree to which you get a delta of the cross-sectional area with the sensitivity of it. And the prediction is that if you want a channel that is not very sensitive, you get a very tiny one where the delta is small, or at least you start with small. And your cross-section is larger and larger, it becomes more and more sensitive. And the best example of that is obviously piezo. Piezo is an enormous footprint, and it's one of the most sensitive mechanosensitive channels we know. It's responsible for soft touch in the skin and a bunch of other things. So you can modulate that given that basic principle. But the fact is that all of these channels are evolving in the background of having to interact with the membrane. And so all of them are what we now in the field call force-form-Lipid mechanism. The way each one solves the problem is a completely different game, because as you can see, the four major examples, one I didn't show the structure, but let's say TREC, Piezo, MSCL, and ICS have essentially orthogonal structure. They have nothing to do with each other. You could not pick one in regards to the other. So there are different ways of solving these particular problems. But the fact is that they are, at their core, what they do is they sense the physical state of the body.
Mm. Okay.
Okay, fantastic. Well, I'm glad you. I'm glad that you allowed a lot of time for questions. And
Oh, absolutely, no question. Yeah.
this was a fantastic talk. I think it was a lot of fun, at least at this end. I hope it was at that end too. And we really enjoyed having you there.
Very well.
End the meeting now, and thank you again.
It's been my pleasure. Thank you very much, and thank you everybody for joining.